hot topics in biochemistry and chemical biology these days is the idea of targeted protein degradation, such as with PROTACs or IMIDs, so like immunomodulatory drugs, molecular glues. And then we have the flip side, we have targeted protein stabilizations with things like DUBTACs. So what are all these talking about? They're ways in which we can take advantage of and like hijack our cells' own machinery that our cells use in order to target proteins for degradation or save them from being degraded, and actually tell that system, okay, well, degrade them now and don't degrade this now. And we can do this for specific proteins using these various molecules. And so today I want to tell you more about how these work, as well as some of the nitty-gritty about why it might not be as easy as it sounds. The basic idea is that we're taking advantage of a cellular machinery of the Ubiquitin-Proteasome System. So basically, when a cell wants to get rid of a protein, they put this tag on it called ubiquitin. It's this little protein. Then it gets this chain of ubiquitin tags that serves as a signal. It's like a direct tag to get to the proteasome, which is like a protein chewer upper. So your cell typically decides when and where it wants to put those little ubiquitin tags. With targeted protein degradation and non-degradation stabilization, what you're doing is you're saying either put a tag there or don't put a tag there on a specific protein. So you're dictating which proteins are getting degraded when. How do you do this? You have to basically get the ubiquitin ligase, so the thing that sticks on that ubiquitin tag, to want to bind to your substrate, or you need to, in the case of stabilization, take those ubiquitin tags off by getting a deubiquitinase to bind to your substrate. So the substrate, the thing that's going to get modified, the protein of interest. And so there are a couple of main strategies with the targeted degradation. We can talk about PROTACs, which are proteolysis targeting chimeras. So they're chimeras because they're heterobifunctional, as I'll get into. They have this one part that's going to bind to your protein of interest, one part that's going to bind to that ligase, so that ubiquitin-adder. And this is going to bring them together, even if they wouldn't normally be brought together. Now, once they're brought together, you can get the ubiquitination and subsequent degradation. In the case of a molecular glue, here instead of having like a chimera, you have one molecule that's going to bind to both things at once. So it binds to the ligase and to your molecule of interest, or it binds to the molecule of interest in such a way that it changes the shape of that molecule of interest so that it will bind to the ligase. And so these are typically smaller, and they're like one part, as opposed to the PROTACs, which have these two parts and then a linker region. And then finally, so an example of one of those would be like thalidomide, and I'll get much more into it later. And then in the case of the targeted protein stabilization, here we're talking about like DUBTACs, so deubiquitinases targeting chimeras. Same sort of idea as with the PROTACs, where you have those two parts, but here instead of targeting the ligase, one of those parts is actually going to bind to a deubiquitinase. So an enzyme that actually removes the ubiquitin tags, rescuing it before it actually gets to the proteasome. And so they all have different uses, they all have different pluses, cons, challenges, and so here's a little more detail about how it all goes down, hopefully.
Our bodies use the UPS, not the shipping company, but the Ubiquitin-Proteasome System as a way to get rid of damaged or otherwise unwanted proteins. But they're deciding what is unwanted. What if we want to decide what's unwanted and what's wanted? Well, we can hijack this UPS system in order to control the levels of proteins to protect the ones that we want to keep protected from degradation using targeted protein stabilization, TPS, such as with DUBTACs. And we can get the ones that we don't want anymore, maybe they're causing problems, we can degrade those using targeted protein degradation, such as with PROTACs or with molecular glues. And at least this is in theory, as I'm going to get into, it's a lot harder than it sounds. And there are a variety of issues that can happen, as well as a variety of new avenues that this field is going. So it's definitely something to keep an eye out for. I was running across so many papers and things about this that I figured I should do a post. So I'll get much more into the details later, but let's just talk about a basic overview. So as I mentioned, we're going to be hijacking this UPS, and this UPS is a normal thing that our cells have, but they're deciding what they want, when and what they want to use it on. So these ubiquitin ligases, these is what like at the heart of this ubiquitin-proteasome system. Their key components are going to be your ubiquitin ligase, which is going to add the ubiquitin, and the proteasome, which is going to chew it up. It's going to chew it up because you've added that ubiquitin. So ubiquitin is just this little protein that gets added onto the ends of these proteins, to the other proteins or onto another site on the protein. That's going to be a key thing, is that this ubiquitin gets added to like a lysine residue, so one of the protein letters. And so in order to actually get ubiquitinated, there has to be like an accessible lysine, or the N terminus of a protein. So it could be the N terminus or the very end of a protein. All proteins have one of those, or it can be a lysine somewhere else. But this has to be accessible to the ligase when the ligase binds to the substrate, so to the protein that you want to get degraded. The way that the ligase binds to that is it has like a substrate adapter. It has a part that can bind to the substrate, so to the protein of interest, and target it for degradation. It also has other parts. So the ubiquitin ligases, there's multiple kinds of them, but they have multiple parts that do different things. And the part that we are focused most about is going to be like this E3 part, this part that actually is going to bind to your substrate. Turns out that there's like about 600 or so E3 ligases in our body, but we typically when designing these sorts of drugs, they're only using a couple of them. So the couple that get used the most often are going to be cereblon, CRBN, and it's going to act as part of a colon 4A E3 ligase complex with the DNA binding protein DBB1 and RBX1. And don't worry about the DNA binding, that's just part of the name. It must be how they discovered it or something. The other one that's commonly used is VHL, von Hippel-Lindau, and it acts as part of this CRL2, so another colon ring ligase, VHL E3 ligase complex with the longin B and C, colon 2, so col2, and ring box protein 1, RBX2. So much more than you probably wanted to know, but if you did want to know, there's the information, and these ligases look something like this. So we're just going to focus on thinking of them as like two main parts. We have the part that basically is doing the ubiquitin, or it's basically just like the core of the ubiquitin ligase, the part that's going to be like the same for these different, for all of these different things. And then we have this substrate part, that's the substrate binding part, this adapter part. And this part can be different, but it's typically going to be either that, at least for now, it's typically going to be that cereblon or the VHL. In order to, so these adapters can naturally recognize various features of proteins. So different ones of these substrate adapters will recognize different like degrons, these degradation signals. So these can be simple things, these can be more complex things, they can be modifications. Various things can act as degrons, and much more on that post in the ubiquitin-proteasome system. So for example, a recent article came out that was actually pretty cool. One of the motivations for this post was that they actually found that the C terminal, that's the cereblon, so the CRBN, the one I was just talking about, it can actually like recognize C terminal cyclic amides, which sounds really scary, but basically it can happen when a glutamine or an asparagine actually like attacks the backbone of the protein and causes it to like cuts the chain and causes this cyclical formation. And you can also get it at the ends of proteins. And so this can potentially serve as a signal for like a damaged protein or something like that, and then allow it to be bound by the cereblon and degraded. But as I'll be talking about, the reason why this was like a cool finding was that people didn't really know exactly what was how cereblon naturally determined its substrates. The way that we take advantage of it most of the time is by using it in combination with an adapter, with one of these like molecules that can allow it to bind to other substrates. So by fusing a substrate to something like thalidomide or lenalidomide, these things that are known to bind to cereblon. And then you can make it something that new, get degraded. And so these thalidomide and lenalidomide, let me step back and tell you a little bit about these. So these are actually examples of what we call molecular glues. So I'm going to be talking most about PROTACs, but it's also important to mention that there's this other class of TPD, this molecular glue. So let's get a little into the details and the distinctions. So in both of these cases, our goal is to get something that wouldn't normally be targeted by the proteasome, or at least not strongly, in the case of molecular glues, and to strengthen that interaction, a weak interaction, or to make a new interaction. So to generate like a new substrate, so a neosubstrate, to make something a neosubstrate. So we have some sort of protein going about doing its business, acting as normal, maybe acting more than we want it to do, and potentially causing problems. Now we're going to add some sort of drug. This can be a PROTAC or a molecular glue. This drug is then going to cause that molecule to be bound by the ubiquitin ligase. And once it's bound by the ligase, it's hopefully going to get ubiquitinated. And then once it's got that ubiquitin tag, it can get targeted to the proteasome and get degraded. So there are basically two main strategies to go about this in terms of the actual drugs. One is a PROTAC and one is a molecular glue. So PROTACs, or proteolysis targeting chimeras, are what we call heterobifunctional molecules. So they have two different hetero parts that do different functional things. So there's the functional. So in this case, one part is going to bind to the ubiquitin ligase, one part is going to bind to the protein of interest, and they're going to be connected by a linker. And so you can swap out this part that's binding the protein of interest. If you have different proteins of interest, you can swap out that part. Then this other part is going to be specific for that ligase, so you can use that same part that's specific to that ligase by targeting multiple different proteins. And so in theory, if you have a molecule that can bind to a specific protein, you can then attach it to this molecule that can bind to the ligase. This will allow them to be brought together, and you form this ternary complex. So where you have these three things, you have your protein of interest, the ligase, and the molecule, and they're brought together. You get a functional complex. The ligase ubiquitinates the POI, the protein of interest, and it gets degraded. And so this is, at least in theory, and as I'll get into, there's challenges because it could be hard to form an actually functional complex. You have to have a site that's available to actually get modified, et cetera, not to mention that these molecules have to be able to get into the body and into the cells that need them. So I hear a lot of talks and things about PROTACs, and people discussing these various issues that make it harder than it seems. But it is definitely an area that is of high interest and promise, both like in the lab setting where it's a little easier if you don't have to worry about getting into bodies and stuff, but also in the clinic for targeting basically anything. One of the benefits is that it's able to target sites on a protein other than the active site, so other than the sites where the things are actually happening. And you need lower concentrations, since a single copy of this can induce the degradation of multiple proteins. because the protein is getting degraded, but not this actual drug. And so this drug can be used over and over and over again inside of the cells, making it a lot better than just a typical drug compound that has to just sit around there, even though it's not really doing anything. It's just keeping the protein from doing things, but it's just stuck in there. So we'll get much more into this, but first I just wanted to go back to this idea of a molecular glue. So as I mentioned, thalidomide is an example of a molecular glue, but we can also use it by like fusing it because it's recognized by the cereblon. We can fuse it to other molecules as part of a PROTAC. But on its own, it can act to bring together molecules and make neosubstrates. So make things that weren't substrates before become substrates. And remember, substrate is just something that's going to get acted on by the ligase. The case of a molecular glue, here you have a single part, and that single part is going to bind to both the protein of interest and to the ligase, but in like a single part, so you don't have like two separate parts of the linker, as in the case of a PROTAC. Instead, you have a single thing. Some of these also, or in addition, they can change the shape of the substrate, so of the protein of interest, to make it so that now it wants to bind. And it can, in addition to making things a new substrate, it can also strengthen weak interactions that were already present. The problem with these is that they can be really hard to predict what they're actually going to do, and like what they're actually going to target. So you've probably heard of thalidomide in terms of its being like a tetratogen, so it can cause birth defects, and this caused a lot of problems when it was being given as like an anti-morning sickness pill to pregnant women, and then lots of babies were born with birth defects. It was discovered that this compound can actually bind to and cause the degradation of various important molecules, such as various transcription factors and things like that. So there's less work done in molecular glues, in part because they are hard to find and hard to predict. So the cases that have been found have mainly been found by accident. There are some that have been found in like high throughput screens. There are some that are beginning to be designed more rationally, but it's really hard because you have this little molecule, and as in the case of the PROTACs, you're actually able to kind of separately design the different parts and put them together. Although the putting them together, well, now you're increasing bulk, you're making it harder for those to actually get into the cells. You have to optimize the linker and things. I'll get much more into this. But it's kind of easier to rationally make than in the case of one of these molecular glues. But there is effort being done on it, in part because these molecules, because they're small, they're going to be potentially better drug targets or drug candidates. And so more on this in this recent article that I will post a link to, but you can see how it's kind of inducing these two molecules to come together. And now this CK1 alpha, which by the way is a protein that I studied in undergrad, or not undergrad, why did I say undergrad? In grad school, it actually, lenalidomide, will get it to bind to cereblon, and then you get the degradation. So that could be one cause of the side effects as well. For like lenalidomide. But these drugs do have important functions still. You might have heard sometimes of these as IMIDs, so immunomodulatory drugs, because some of the targets that they actually target have immune functions, and so these drugs are actually able to help with some conditions. But yeah, don't you take them if you're pregnant. And yes, so I'll post a link to that. I'm going to go back and talk more about the PROTACs. This is what you hear most about because you have that better specificity, I guess, at least in theory, and easier design. And so here are some examples of what these look like. And so here you can see that you have this molecule that's binding to the substrate, and it's binding to and it's binding to the ligase, and you have this kind of these two different parts with this linker in between, as opposed to the case of the molecular glue where we had this one little molecule that's kind of just making these stick together. I mentioned there are a number of benefits, and so I want to go in a little more detail about what some of these benefits are. And I know I keep saying like, oh, there's all these difficulties, but the reason why people are still pursuing it is because it has such potential, and there are examples where it can work really well. One of the reasons why it has such potential is because you can target sites other than the active sites. So traditionally, drugs, if you want to stop a protein from doing what it does, you would target this place on that protein where it does what it does. So say an enzyme, a reaction mediator or speed upper, there's going to be like a pocket on that enzyme, the place where it like brings together different components, so it binds to things or binds to like a single substrate and then changes it somehow. And so we call this place on the protein the active site. So there are a lot of different enzymes, and there are a lot of enzymes that are specialized to do specific reactions, but that specialization comes from like multiple aspects of the enzyme and not just that one active site. That one active site is actually generally pretty like generic in terms of, say you have talking about kinases. Well, kinases, they add these phosphate groups to proteins, and they typically do it using this like same conserved mechanism. Because it has that same mechanism at the heart of its active site, it's all going to be the same. And so if you want to target that very core of the active site, well, now it's going to be hard to specifically target it without targeting all of those other kinases as well. And so in order to get better specificity, people have been trying to find all sorts of alternative strategies, because if you just try to target that active site, well, now you're going to get a lot of off-target effects. But if you could target something other than an active site, well, then you have a better chance of getting better specificity. The reason why sites other than active sites aren't typically chosen is because, well, if you're not targeting the active site, well, then unless you're causing some sort of conformational change, so some sort of shape change that makes it so that the protein is no longer active, even though you're not like inhibiting the heart of it, then you can cause inhibition. But this is not as likely. And the sites other than the active site can traditionally be kind of like harder to bind to. And also because we can target non-active sites, we can then target proteins that like don't really have active sites. Maybe they have a site where they bind something, but they don't really have an active site, or other things like that. And you can kind of just, if you have a known like inhibitor, or you have a known, if you have a known binding partner, you can use that as a starting point for making a PROTAC by hooking it up to a part that's going to bind to the ligase. Sometimes you even start with an inhibitor. I know it, or like an active site inhibitor. And so I know I've been mentioning how one of the great things was that you can use it for things other than active sites, but it can still be better than a traditional active site binder in the sense that you don't need to use as much of it because they're able to act substoichiometrically. So stoichiometry refers to like the number of copies of one thing compared to another thing. If you think about a traditional drug, this is going to have to act as in like a stoichiometric manner, where you'd have one copy of the drug bound to one copy of the protein at all times. And so if that drug comes out, well, now that protein's active again. In the case of these PROTACs or the molecular glues, what happens is that you can act substoichiometrically, meaning that a single copy can lead to the degradation or to the stabilization of multiple copies of the target. And so you have this one copy binds, and then you have a single copy that binds, then the protein that recruits the ligase or recruits the deubiquitinase, it does its thing, so it ubiquitinates the protein or it deubiquitinates the protein. And I'll talk mostly about the degradation. That's what we're most, DUBTACs are a little newer and not as well studied, but in the case of the degradation, you get like the ubiquitination and then the subsequent degradation. And now, once that protein is degraded, well, this drug is still good, this ligase is still good, and so it can act on multiple proteins, and you're able to get more bang for your buck instead of having to have that drug sitting around. And because it doesn't have to stick around as long, you can also use lower concentrations just because you don't need that binding to be like as super duper strong because it doesn't have to stay around as long. Okay, so it sounds really, really great, right? But there are some hurdles. So it can be difficult to design a heterobifunctional compound. It's not just like you're taking those two pieces and just sticking them straight together. If you think about that scene in Lady and the Tramp where they have like that shared spaghetti, well, they get that spaghetti pretty short and then they kiss. They go, yeah, and kiss. So, but say if you went even shorter than that, well, you couldn't. Their teeth would stick together. So you can't just stick these molecules together because there's not then room for them to bind. But if you lengthen that linker, well, now there's more room, longer spaghetti. Now they can get to a comfortable kiss. The problem is, as you expand that linker, well, now you're adding bulk to the molecule, and you're often adding like hydrophobic E regions. So you're basically potentially making the molecule harder to actually get into cells, which can be a problem if you're trying to use this to treat, to like stop drug proteins or from being degraded or not degraded inside of cells. And so the solubility issue can be a main hurdle, even if you get an initial like hit with just like if you're just putting them in a mix of molecules. Another problem. Okay, so even if we get the complex forms, if we get that ternary complex where we have I'm just going to talk in terms of the ligase. It's easier, but also the ubiquitinase, the same thing. You need to have the protein, the drug, and that ligase be in a ternary complex. So the three parts of them, but you need to be active and you need it to be like active where you want it. And so basically you need to have it so that when they're bound, there's going to be a site nearby that can act as the ubiquitination spot. So the ubiquitination can happen on the N terminus of a protein, so the very end of a protein, or it can happen at a lysine residue, so the amino acid lysine. And that lysine, it can't just be like, if it's hidden in the protein, that's not going to be helpful. It has to be somewhere kind of flexible where the ligation, where the ubiquitin can get added. So the site can't just be like anything on the protein. And sometimes drugs can form these complexes, but not actively, not form like functional complexes that are actually going to ubiquitinate the protein. And without that ubiquitination, then they can't get degraded. The activity can also vary depending on the subcellular localization of the target, as well as based on that target itself. So how well that target can get ubiquitinated or trafficked to the proteasome. The actual location of that protein within the cell can affect things. And so here was a recent article in Cell Chem Biology where they were showing that where the PROTAC was, where the protein was, all of this mattered inside of the cells, but it wasn't like you could say, okay, well, if it's in here, then it works well. If it's in there, it doesn't work well. It also depended on the protein. So basically, it's complicated. And another hurdle. So this has been another benefit of this is that it has a reduced, like, or at least in theory, reduced resistance capability in terms of cells or like cancers, there's developing resistance to it. But resistance is possible, such as with mutations in the E3 ligase that's being recruited and or other components of the UPS. But because these components are also used by the cells for the normal stuff, and hopefully the resistance will be less likely. In terms of future places to keep an eye out and to further reduce the off-target effects, you can try to restrict the activity more. So even if you are not able to make a perfectly 100% specific PROTAC in terms of what it's targeting, if you can restrict its activity to where you want it to target, so to like a tumor, say, well, then you might have better luck, or to a certain cell type at least. And so an area of future development is to try to do this more like tissue and cell specific targeting. For a lot of drugs, what is done is that the targeting mechanism is done in like trying to take advantage of the uptake. So maybe coat the drug in something that is going to then bind to the surface receptor that's only found on these cells and take it in and that sort of thing. But with this strategy, what you can do with these PROTACs is you can actually get them, in theory, to use a ligase that is predominantly expressed in just a single cell type or just that single tissue type that you're interested in. So now your drug, even if it gets into all of the cells, it's only going to be active in those cells that have lots of that ligase acting, and this can then allow you to use it. So you would think that with like over 600 of these E3 ligases, then you would have lots of opportunities to do this. However, there's a reason why only a couple of them are typically used. They tend to have a better chance of working. And so here was another recent article from Cellcom Bio, where they were looking at using a different E3 ligase, this KEAP1, and they found that, well, it didn't work that great. It was able to degrade some substrates, but not others. And it wasn't able to degrade substrates that they showed they could degrade when they use VHL or Cereblon. And so basically, they outline some difficulties of trying to use like non-conventional E3 ligases as the thing. But all hope is not lost. Here is a recent preprint on proteasome scale-induced proximity screens used to try to find alternative ligases and deubiquitinases and things that can be used. They found too that there was a wide variety of their efficacy against different substrates and things like that. So basically, it's complicated, but there is a lot out there that could be tapped, a lot of potential that could be tapped. But at least until more is understood, it's probably going to involve for all of these a lot of just like screening and trialing to try to find things that work. Here is another a recent review that was nice. This is where those structure pictures came from before, and it has information about various compounds, as well as if you want to look at more of the structures, it has the PBD codes and things, which I find could be helpful. Other avenues, so I've been talking a lot about PROTACs, but there are other TACs. So these include autophagy targeting chimeras, so these AUTACs, and lysosome targeting chimeras, LITACs, so basically taking advantage of other cellular destruction machinery. LITACs, these were actually developed by Carolyn Bertozzi, who recently won the Nobel Prize, by the way, for his glycobiology work. And these LITACs, they have the benefit of being able to target and degrade membrane-bound and extracellular proteins. They have this this like glycan, so that's how it ties in with her work. This glycan that is a ligand, so it binds to this receptor on cells that this receptor then gets trafficked into it's like makes a lysosome, so this internal cellular component where degradation can occur. And by putting that target onto something, by putting that glycan onto a molecule that binds to your target, then you can then traffic your target to the lysosome. So pretty cool stuff. And I didn't talk about it that much, but targeted protein stabilization, so TPS, the flip side of the coin, where we're trying to prevent a protein that's been ubiquitinated from getting degraded. This can actually be good if you have protein that's getting made, but maybe it's not perfectly functional. Maybe it has some sort of problem because there's a mutation in the gene, say, but you want to keep it. You don't like it's good enough to do some stuff, and so you want to keep it around. Or maybe it's getting degraded or ubiquitinated for who knows why. But anyway, you want more of it. You want to keep it around, and so you could let it survive by removing that ubiquitin before it actually gets to the proteasome. And so this is the idea behind TPS and DUBTACs, so deubiquitinase targeting chimeras, targeting these proteins that are actually going to remove the ubiquitin. Speaking of these dubs, they can be another reason why your PROTACs might not be working if they're getting counteracted by the DUBTAC, by the deubiquitinases, because these are naturally found in our bodies, because our bodies are really good at controlling the levels of the ubiquitination so that we don't degrade things when we don't want to, and we degrade things when we do want to. And the idea with these targeted approaches are that we're actually telling it when we want to degrade and not degrade specific proteins. So hope that helps you understand them. And yeah, so you'll probably be hearing a lot about them lately in the future. And so I just wanted to kind of fill you guys in.